talking to me?
Yeah.
My real life?
Yeah.
That's
I
not
mean, I bought these. My husband and I bought these shirts, and I thought I'm never going to wear that for anything. And then I thought, oh, I should wear that tonight.
Look at her.
I'm pretending.
Oh, and pumpkins.
Are you pretending or are you really?
Or are you really a hippie?
What's the RNA?
Suspicious.
My children always thought that was.
We told you this. This is the third time I've told you today. This is the third time I told you in like an hour.
There's so much homework.
Does it have a
I
link?
literally, it's the third time.
Yeah.
That's the third time you've said that
I'm
too.
pretty sure it was.
Can you just stick to
I'm pretty sure she put it in.
Oh my god.
So we didn't quite finish this topic, so I thought, oh, we're going to finish this topic before we rush on to the next one. So we're still talking about how to mutate. We went through a variety of chemical factors. Actually, we went through very few because there's way more that could have been covered. But we're going to jump now to physical factors that can be recognized as mutagens. And again, we're only looking at a couple of different things here. So ultraviolet light is one that is well recognized, and because it's something that is always present, because it's electromagnetic radiation from the sun, then lots of organisms are exposed to it, and people have lots of opportunities to see what exactly does it do. Ultraviolet light causes damage to DNA by causing the formation of dimers, and although it says it's dimers between pyrimidine bases, I've only ever seen it described as thymine-thymine dimers. So what happens is two thymines bond to one another. Consequently, when the DNA replicates, they don't behave properly. So here you see two thymines. They have to be adjacent to one another. Ultraviolet light is going to cause them to stick to each other, forming a dimer. The term dimer just means think about monomer, polymer. Dimer just means two units, two of something, in this case, two thymines. Now, if those two thymines are stuck to each other, and if everything else was the same in terms of how transcription happened, would you expect transcription to go ahead? And if so, what kind of mutation is this going to cause? Or if this DNA replicates and the complementary DNA sees that thymine is one instead of two, you're going to get a deletion type point mutation. So thymine-thymine dimers could potentially cause a deletion, and remember, a deletion is like an insertion. You're going to get a frame shift, a shift in the codon reading frame, so it's going to change or potentially change all the amino acids beyond the point of mutation. So we see the DNA up above with the two thymines that, in this case, what is circled, these two thymines are coding for only one adenine instead of two adenines, and so what would have been the sequence with two adenines, you'd have glutamine here. When there's only one adenine, then we have a change in the amino acid beyond that point. What the mutation occurred here. So this one changes, but this one and whatever comes after changes also. Now, because prokaryotic cells do polycistronic transcription, if you have a mutation like this that's up toward the front end of the operon, it's not going to change one gene. It's going to change a whole series of genes. And because we know that most enzymatic pathways involve enzymes that are constitutive, they're essential to cell survival, so usually this type of mutation is going to be useful. Consequently, ultraviolet light does tend to kill bacteria. The other thing that can happen when these thymine-thymine dimers form, if this happens and then the cell is trying to replicate that DNA, sometimes it causes DNA polymerase to fall off, and so it terminates replication. They simply can't replicate properly. Or if they were trying to transcribe, they won't transcribe properly either. Either way, thymine-thymine dimers tend to be lethal to cells. And so ultraviolet light is used in a variety of settings to control bacteria. So you can see some of these are clinical settings. We're going to be doing some experiments in the laboratory where you get to see that ultraviolet light exposure is definitely lethal to bacteria, but it has to be a direct exposure. It doesn't have real good penetrating ability, so if there's anything in the way, it doesn't do much. Ultraviolet light is also being used in water treatment locally because they're trying to get away from using chlorine to treat water, because if they put chlorine with water after it goes through the wastewater treatment facility, then they have to take the chlorine out before that water can go into a stream. So if they don't put the chlorine in, they can use ultraviolet light instead, and it seems to work fairly well. Again, the problem is the water really has to be fairly clear. If the water's really murky, and that's what happens in the wintertime when you have a lot of sediment in it, then ultraviolet light's not going to work. So it has to be a pretty direct exposure. Now the other thing that happens, because so many bacteria are exposed to ultraviolet light, many of them have developed ways of dealing with this type of mutation. So they've developed resistance to ultraviolet light. It's not that they aren't damaged by the DNA, but they have devised mechanisms by which they can repair that damage. And they have two different types. So some bacteria use what's called light repair. Light repair involves enzymes that are actually activated by exposure to visible light. And if they have this light repair, what they'll do is find the thymine-thymine dimer and just take it apart. So the DNA, the damaged strand where the two thymines are stuck together, those two thymines come apart, and then that DNA is going to be back to normal. There are also bacteria that use enzymes involved in dark repair. This is a little more complex. If they use dark repair enzymes, they don't require dark, they just don't require light. And what they will do is recognize a portion of the DNA that's damaged, and they'll cut the DNA, the damaged strand, and take out a section, removing the damaged part where the dimer is, and then that complementary strand will code for a new strand of DNA, and they'll put it back properly. There are enzymes associated with, well, DNA polymerase. We talked about DNA polymerase 3 being the primary builder. The other DNA polymerase portions are involved in proofreading. So they run back and forth on the DNA strand to make sure that it's being properly synthesized. And if it isn't, they have some ability to fix it. Another type of mutagenic agent is ionizing radiation. But ionizing radiation, which would be X-ray, gamma ray, this sort of thing, these can cause very severe types of damage, and they tend to be lethal to cells, not just prokaryotic cells, but eukaryotic cells as well. So these can damage individual bases, they can cause breaks in the DNA, they can cause chromosome rearrangements, they can cause cross-linking across the strand. Again, if you have covalent bonding across the strand, then the DNA can't replicate and it can't be transcribed. So again, ionizing radiation is lethal, but even here, there are some bacteria that are resistant to exposure to ionizing radiation. So I mentioned these guys early in the semester, Deinococcus radiodurans. Sometimes these are referred to as Conan the bacterium. They are exceedingly resistant to exposure to radiation. There's some thought about why would they have evolved this ability, because apparently they are resistant to much greater exposure to radiation than they would ever be exposed to on the planet's surface. So researchers aren't sure why they're like this, but again, they don't stop the damage from occurring. They have enzymes that fix it. So when ionizing radiation causes damage to their DNA, their enzymes can then fix that. Okay, so that's the end. Again, that's a real short presentation on mutagens, simply because we don't have time. Introductory class, just bouncing rocks off the surface. Always bouncing rocks off the surface. So now we're going to be looking at mechanisms of genetic exchange, also referred to as horizontal gene transfer. So, genome, total DNA content of the chromosomes of the organism. The genome, that's all the DNA associated with the chromosome. So genome includes not only structural genes, but promoters, operators, intergenic regions, everything. But anyway, the genome, the content, is definitely going to influence the genotype. Genotype is genetic potential. What type of characteristics could the organism have were its genes expressed? These things are not absolutely static. The genome, consequently the genotype, can change over time, and we've looked at one way. So one way change occurs, of course, is mutation. We just got through going through this section on mutation. Another way that the genome can change is through what's called genetic exchange or horizontal gene transfer. So cells can transfer DNA from one to another. In eukaryotic organisms, the reorganization of genetic material occurs during sexual reproduction. So you have zygotes that are genetically a little bit different. Those two zygotes fuse, the gametes fuse and you get a zygote. They're genetically different, and consequently you get genetically unique offspring. So again, in this diagram, we've got purple organisms and orange organisms, and then we get a wide variety of offspring there. None of them are quite like the ones that we started with. This is advantageous to organisms because there's always selective pressure in the environment. Organisms tend to produce way more offspring that are likely to survive, and the selective pressures will eliminate some of them. The ones that get to survive are potentially better adapted to that environment. But then the environments are always changing too, so this is an ongoing shift. For a long time, biologists thought bacteria didn't undergo sexual reproduction because they noticed that bacterial cells never fuse to form a zygote. You don't have sex cells, gametes, coming together. So it was assumed that bacteria actually don't reproduce sexually. We now know that they do. So the way they do it is just a little different. They have mechanisms that are unlike the mechanisms we use, but they still do it. There are 3 types of genetic exchange or horizontal gene transfer that are recognized to occur within bacteria. Anytime I say this and I think, well, okay, there are 3 types, 3 mechanisms that we know about. Could there be something else? Maybe. We just don't know that much about bacteria. But anyway, these 3 mechanisms all have some features in common, and we're going to talk about the things they have in common and then talk about these mechanisms. So the 3 mechanisms of genetic exchange for horizontal gene transfer are called transformation, conjugation, and transduction. Remember I told you guys watch out for the trans words? Get transcription, translation, transposons, transposase. Now we've got transformation and transduction. Oh my word. Watch out for the trans words. So again, before we look at how these mechanisms differ, we're going to look at how are they all similar to one another. First of all, when you're dealing with bacteria, and as far as I know this is true of archaea also, the cells don't fuse. You never have the joining of 2 cells to form a zygote. So you don't have haploid cells coming together to form a diploid cell. They just don't do that. So there's no zygote formation. Second thing about genetic exchange with bacteria, and presumably archaea, is it's one way. So one cell donates DNA, the other is just a recipient. You don't have DNA going back the other way. We use the term conjugation with bacteria, but it's not the same as conjugation with paramecium. When paramecium conjugate, you have 2 cells. They both donate and receive DNA, and bacteria don't. It's a one-way exchange. One is a donor, the other is a recipient. The other thing about the amount of DNA transferred, it's typically not the total complement of the chromosomes of the cell. My understanding of paramecium, again, is they have multiple chromosomes in these little micronuclei, and they send multiple chromosomes back and forth. When when bacteria transfer DNA, it's typically a very small amount. It's only a few genes. There have been some circumstances where Escherichia coli has been forced to transfer the entire chromosome, but they only do this under experimental conditions, and I'm not sure how they make them stay together. Really? You tie them up with a rubber band or what? I don't know. Anyway, so usually it's only a few genes that get transferred. The other thing that happens is typically the DNA that is transferred will enter into the chromosome and it'll replace homologous DNA that's already there. So again, you need to be familiar with homologous DNA. I gave you this term last time, but hey, repetition is learning, right? So it's always good to review. What are homologous genes? What does that word mean? Same, but yeah, not actually. So homologous DNA is DNA that encodes the same characteristic in general, but it's not identical. So let's say you have a gene encoding for flagellin protein, and some of the flagellin proteins are purple and some are pink and some are green. Different genes have slightly different amino acid sequences. They all encode flagellin protein, but they're not identical. Those alternate forms of genes are called alleles. So alleles are alternate forms of genes. They're homologous DNA. Something's different about them, different base sequences. I don't know if that bothers you. We should have drank 3 minutes. So another feature that these exchange mechanisms have in common, they seem to be most successful at the bacteria that are exchanging DNA are closely related. Of course, humans get around this because we know what's involved in this relationship business. The reason that bacteria have to be fairly closely related is because bacteria contain enzymes called restriction endonucleases. The term restriction has to do with them restricting random genetic exchange. But if you think about it, again, as I was mentioning with mutation, if bacteria could change, bacteria undergo mutation once a day. If all of those mutations were kept, you wouldn't be able to have bacteria that had any consistency. How could you work with Escherichia coli today and work with Escherichia coli again next year and expect it to be the same? Most mutations are deleterious, and most genetic exchange is blocked by restriction enzymes. So these restriction enzymes or restriction endonucleases, basically what they do is recognize DNA that's not part of the cell and chop it up. They destroy it. We'll talk a little bit in lab about what makes DNA foreign versus cellular DNA. How do they recognize it? Okay, so the other thing that is true of all these genetic exchange mechanisms, if they're successful, if the recipient takes in DNA and is able to incorporate it into its chromosome and hold it there, then we have what's called recombinant DNA. This term, unfortunately, recombinant DNA technology has to do with any kind of manipulation of cells where you're taking DNA out, putting DNA in. Even what we did for processing cells to run the PCR, that's potentially recombinant DNA technology, even though we didn't generate recombinant DNA. But recombinant DNA technically is a combination of chromosomal DNA from more than one source. It happens naturally, but humans do it a lot because now we can take genes from any source and put them into E. coli or put them into yeast cells to get those cells to do what we want them to do. So now let's take a look at the three mechanisms of genetic exchange or horizontal gene transfer. The first one is called transformation. This is also sometimes called DNA-mediated transformation because technically it doesn't require that the donor cell be present at all. The donor cells simply have to donate DNA, but the DNA could be loose in the environment and the cell's not there. So another way to think about transformation, it's just a mechanism that allows bacteria to pick up naked DNA from their environment. Well, that brings up the question, then what am I talking about when I say naked DNA? Any guesses? Well, it's not in a cell, but it's also not surrounded by a protein coat. If you put a protein coat around DNA, then you have a virus. So transformation is not involving virus. It's just DNA coming from a donor cell, but that donor cell might be dead and no longer in the picture. In fact, we're going to do transformation in the laboratory, and the donor cells are in this flask. Well, the remains of them. So if you remember all the mean things we did to the bacteria last time, where you beat them and treated them with various types of chemicals, and ultimately we threw their dead bodies in this flask. The DNA from these cells is in the freezer. It's all by itself. And when we get ready to do transformation, it's DNA mediated. The DNA is just there and the cells pick it up. So the donors are not only dead, they can be completely gone. So again, naked DNA, not covered by a protein coat. If it has protein, then it's part of a virus particle, which is called a virion. That's a really complex looking virion. So how do we know that this mechanism even takes place? How do we know that transformation occurs? This was discovered by a researcher by the name of Frederick Griffith in about 1928. And what Griffith was doing was studying organisms that were thought to actually he knew they caused pneumonia. I think it was at the time they were trying to figure out what to do with the influenza epidemic also, or this kind of came about after. But anyway, he was working with Streptococcus pneumoniae. My thought is when you look at what kinds of things he was doing, he was probably working on vaccine development. So Griffith knew some things about Streptococcus pneumoniae. He knew that if you put Streptococcus pneumoniae into mice, if they were live Streptococcus pneumoniae, and if they were capsule forming, those were the virulent forms of Streptococcus pneumoniae. So if you put those into mice, the mice got pneumonia and they died. On the other hand, you could use non-capsule forming Streptococcus, and even if they were live, if you put those into mice, they didn't kill the mice. The mice lived fine, or at least they lived fine until the next injection. That was probably a different part of the test. So here's a question for you. Why? What's the difference between capsule forming strep and non-capsule forming strep? Do you remember what capsules are for? Protection. So one of the things, capsule is for, it's a nutrient store, it's used for attachment, but a major factor of capsule when you're talking about disease, potentially disease-causing agents, the capsule makes bacteria less recognizable to our phagocytic white blood cells. So if they see capsule, they don't see that as something they should consume, so they don't. Therefore, capsule-forming strep can survive and cause disease. If he injected mice with dead capsule-forming strep, again, why would he do this? Well, he was probably developing a vaccine. That's oftentimes what happens. They're either attenuated cells or they're dead cells. You can inoculate mice with dead capsule-forming strep. They won't develop pneumonia. Furthermore, they'll probably be resistant to pneumonia should they be exposed to it at a later date. But if he used a combination of Streptococcus that would not normally cause any damage, so he used the non-capsule forming strep that were living, and he used the dead capsule forming strep. Either one of those alone didn't do anything to the mice. But when he did this in a combination and gave them to the mice got pneumonia and they died. Well, so then Griffith wanted to know why. What the heck? Why was that?
Bringing the margaritas.
I forgot about that, and I heard it rolling around back there.
I made it into kind of like an active fun activity for them.
What does that mean?
So I had made one out from scratch, so they could label the site wherever they want genetic codes.
Oh, we'll figure that out. Yeah, thank you. That's the ice chest that went to Davis this morning. So this morning, all the DNA samples went to Davis in that. And then it came back in the car and I forgot to take it out. Anyway, no big deal. Anyway, so again, why? Why if you use live non-capsule forming strep and dead capsule forming strep, did the mice die? Well, so Griffith knew about Koch's postulates. And so he isolated the bacteria from the dead mice, and he looked to see what was killing them. It turned out it was Streptococcus pneumoniae, live capsule forming. That's not what he had given them. He had given them dead capsule forming strep and live non-capsule forming strep. So he surmised that what had happened is the live strep picked up the genes for capsule formation from the dead strep, and they became live capsule forming strep, which is exactly what happened. So DNA mediated transformation. Again, after Griffith's experiments, then people started looking and they saw that transformation occurred fairly readily in a lot of different types of bacteria. Bacillus, Pseudomonas, Staphylococcus, Haemophilus, Neisseria. We will be transforming Escherichia coli. So in the laboratory, I'm going to talk to you a little bit about some things we have to do to the E. coli cells to get them to be recipients, to get them ready to take in DNA. But basically, you can pretty much get any type of bacteria to take in DNA if you know what to do. And experimenters have done some really bizarre things to put DNA into bacteria. Bacteria are not sensitive to electric current, as in it doesn't damage them. You can have quite high voltages, enough that it would cause you significant harm. It doesn't do anything to bacteria, but you can get voltage to move DNA because it's a charged molecule. And so they can push it into cells using high voltage. They've actually shot cells, shotgun blast type situation with pellets with DNA, put it in that way. Lots of things you can do to get DNA in. What we're going to do in the laboratory is make some E. coli cell competent. You guys get to make competent cells in here. And it just means making cells capable of taking in DNA. Well then, so transformation happens. Again, bacteria can take up genes from some other bacterium and then take up those characteristics. We know it happens probably more commonly within gram-positives, but it certainly happens with lots of different types of bacteria. The next mechanism of exchange or transfer is called conjugation. Again, that term is not restricted to bacteria, but conjugation as it occurs in prokaryotic cells is very different than conjugation as it occurs in paramecium. So again, it's transfer of DNA from a donor to a recipient. Transformation was transfer of DNA from a dead donor to a live recipient. Conjugation is transfer of DNA from a live donor to a live recipient. And the significance of the donor being alive is that the recipient, or excuse me, the donor has to make sex pili. Sex pili are involved in this operation, and they are only produced by live cells. Also, if I ask you about transformation on a quiz, don't tell me that transformation is the transfer of DNA from a live donor to a dead recipient. Because really, what's a dead recipient going to do with it? It's dead. And I've had people tell me that. It's like, no. You just have to think about this a little bit. Okay, so conjugation involves sex pili. Remember, the pilus made of pillin proteins are these long hair-like structures. Actually, they start out long and then they shorten and pull the cells together. Typically, there's only 1 or 2 pili produced per cell, whereas fimbriae are much more numerous and shorter. So you can see the E. coli over here on the left-hand side has a lot of fimbriae, but there's just 1 sex pilus. It used to be thought, because pili are hollow, it used to be thought DNA went through the pilus, that it was somewhat like a straw, and it went through the pilus into the recipient cell. Now they know that doesn't happen. Instead, the cells are pulled close to one another by means of the sex pili, and then the DNA is transferred. Well, so then there are researchers that have studied Escherichia coli extensively. Escherichia coli is recognized as something that undergoes conjugation, and so researchers want to know how they do it, what can they do, and they found out that there are multiple different mating types, and it has to do with which ones produce sex pili and which ones don't. And the terminology gets a little anthropomorphic here, where people think about bacteria and structures bacteria produce and try to kind of superimpose that over onto humans. The fertility factor, F factor, is a plasmid. So we talked about plasmids, and I mentioned R factor plasmids that encode resistance. F factor plasmids carry the genes that encode the formation of sex pili and also the initiation of gene transfer. So if they have the F factor plasmid, then they can potentially transfer DNA. An interesting term that we're going to run into here is the term episome. So a plasmid, I've given you the definition of a plasmid, that's an extra chromosomal loop of DNA outside the chromosome, small but separate from the chromosome. Well, an episome is a plasmid that can go into the chromosome. So it's not necessarily going to stay out, it can get in, and if it can get in, it can get out again. So we're going to see that F factor can move back and forth between the chromosome and being separate. So F factor is a double-stranded DNA loop. It's about 94,000 base pairs in size. It's pretty good size. It has a multitude of different genes on it. It has 2 origins of replication, which is a little bit different. One of those origins, when replication starts, it starts and goes in both directions away from the origin. The other one determines a unidirectional replication. So if replication starts at the one, not the typical origin, but starts at the other one, it only goes in one direction around that loop, which is significant when we start looking at the different mating types of Escherichia coli. So when F factor is outside the chromosome, it replicates like other plasmids. Replication starts at the origin, goes in both directions. If it gets into the chromosome of the host cell, then the second origin of replication takes over, and the chromosome, the plasmid starts replicating in the middle, replicates through the plasmid, and then onto the chromosome, and has to go all the way around the chromosome, and then get the second half of the plasmid. It's difficult to illustrate this, but anyway, it just has a different replication method. The other thing about Escherichia coli is that the mating types can recognize one another, so you never have female type E. coli trying to mate with other females. The transfer always has to be from male to female, so males are donors, females are recipients. The males produce this sex pili, the females don't. Beyond that, they're really not like sexes at all. There's no sexual dimorphism except for the pili formation. So these are the mating types. If an E. coli cell does not carry the F factor plasmid, it can't produce pili, therefore it can't initiate genetic exchange, it's considered female. It's called F-, but an F- cell is a female type cell. If the cell has the F factor, and it's outside, it's separate from the chromosome, then it's called F+. My guess is it could have more than one F factor, but it's not in the chromosome. Because the F factor is an episome, it can go into the chromosome. If it's in the chromosome, then that cell is referred to as a high-frequency recombinant. It's also male. Any cell that has F factor is male. So the high-frequency recombinant, we're going to talk about why it's called this. Basically, it has a very high potential of causing the formation of recombinant DNA when it mates with an F- cell. And then the fourth type, if the F factor was in the chromosome and it gets out, but when it comes out, it takes a little bit of chromosomal DNA with it. Then you've got F factor as a separate plasmid, but it's carrying a piece of chromosomal DNA. It's called F prime. It's also male, but it has some genes that are part of the chromosome now attached to the plasmid. So now we can take a look at results of conjugation. Always males are going to transfer DNA to females. If you have mating between an F- and an F+, the F- is the recipient, the F+ is the donor. The F+, the male cell, donates F factor to the female, and the female becomes male. The original sex change. You'd think if this continued for any length of time, all E. coli would be male. They'd all be F+. But apparently, even though the F factor is a plasmid, and we talked about how plasmids typically reproduce faster than the chromosome, they don't always. So if the chromosome is replicating and for whatever reason the plasmid isn't, then when the cell divides, one cell gets plasmid and the other one doesn't, so one gets back to being female. Anyway, the population doesn't become totally F+. It's just if you mate an F- with an F+, the F+ donates the plasmid and the female becomes male. And F- becomes F+. I think the F+ stays F+ too. If we have mating between an F- and an HFR, the HFR, the DNA is within the chromosome. So usually what happens is while conjugation is happening, the plasmid initiates replication at the origin that initiates a unidirectional replication. So again, a little piece of the plasmid and then part of the chromosome starts being transferred to the recipient cell. But very rarely will the cells stay together long enough for the entire chromosome to be transferred. And so typically what will happen is the recipient gets only part of the F factor and gets part of the chromosome. Again, homologous DNA will replace DNA that's in the cell, and so the recipient will stays female because it didn't get the whole F factor, but it becomes recombinant. So there's a very high probability that it will get enough DNA from the donor that it has a new combination or a combination of chromosomal DNA. And this is why HFR is called high-frequency recombinant. This is just showing single-stranded DNA from HFR that's being transferred, goes into the cell. This is really a fairly complicated process. The DNA goes in, the homologous sections line up, and then the DNA that was transferred will replace both strands on the chromosome. Again, homologous DNA transferred replaces DNA that was already there, but the recipient is going to become recombinant. Now, if we have mating between an F- and an F', The F prime has its plasmid separate, and it has a piece of the chromosome in that plasmid. So when it transfers its DNA to the recipient becomes recombinant, but it also becomes male because the entire F factor is transferred. This is a diagrammatic representation of this mating. Actually, this is HFR and F plus, different versions of these guys mating with F minus cells, and what you get. When you see, or when you have an F prime mating with an F minus, this is sometimes called sexduction. And I think that term is because it's somewhat similar to a process we're going to see that involves viruses. I'm not sure this term is used very much. Sexduction, an F prime is mating with an F minus. The F prime transfers the F factor, which is also carrying a little bit of chromosomal DNA from the original host. Consequently, during sexduction, the recipient is going to become male, and it's also going to be partially diploid. It's going to be diploid for whatever genes were on that plasmid, separate from the chromosome, because it keeps its own chromosome, and it gets the plasmid plus some chromosomes from the original host. So it's male, it's F prime, and it's partially diploid. They used to use HFR to do gene mapping, and I believe in your notes there's this big section about talking about conjugation where they use HFR mated to F minus, and they would allow mating to occur for a certain amount of time, and then put the cells in a blender and then chopped off their sex pili so it would stop the mating process. And they would do this at different time intervals and look to see which genes had been transferred, which made for nice quiz questions, but it really isn't relevant because nobody does gene mapping this way anymore. Now that we have nucleotide sequencing, it's like, forget this. Not going to happen. So never mind. So that was transformation, conjugation. The last process of genetic exchange is transduction. And transduction is transfer of DNA from a donor to a recipient that involves a virus. In most instances, the donor is going to be dead, but I don't think it absolutely has to be. So this would probably make more sense after we've talked about the viruses that infect bacteria, which are bacteriophage. What I'll do is go through transduction now, and then go through it again after we've gone through viruses and had a better understanding of what viruses do. So again, the viruses that infect bacteria are called bacteriophages. The term literally means bacteria eaters. They don't eat bacteria, but if you have bacteria on a plate, you've all seen lawn cultures where you just have a coating of cells all over the surface of the plate. If there are bacteriophage infecting those bacteria, they will make little divots. They look like little depressions where the bacteria are actually dead in certain regions. So it looked like they were being removed or eaten. They weren't being eaten, they were just being killed by the virus. Here we see some, one electron photomicrograph of viruses on the surface of the cell, and then some illustrations of viruses, different types of virus particles. They come in there's lots of different ways. So again, probably in most instances of transduction, the donor cell is dead, because a lot of viruses are cytolytic, which means they kill their host cells, but not all viruses are. Some types of viruses allow the cells to survive and just shed virus particles for a very long period of time. So I don't think it's necessarily has to be live or dead. So with transformation, the donor is dead. With conjugation, the donor has to be alive. With transduction, either or. I don't think it matters. The other thing, we can divide transduction into two categories. There's generalized transduction versus specialized or specific transduction. And this has to do with what state the virus is in before it's engaging in transduction. So generalized transduction, the reason it's called generalized is because any piece of the donor DNA could be transferred. Typically what happens is you have a cell that's infected by virus. The virus takes over cell function, which is what viruses typically do. And in the process, the virus encodes enzymes that chop up the host cell DNA. That DNA, imagine it being chopped into a whole bunch of little pieces. somewhere along the line, some of that DNA gets incorporated with viral DNA, and then part of the virus life cycle involves packaging, putting those DNA pieces inside protein coats called capsids. If cell DNA and viral DNA get packaged up together, then when that goes into the next cell, that cell is going to take up DNA from a different source. If it incorporates into the chromosome, then it becomes a recombinant cell. So because it's any piece of DNA, it's generalized. Generalized transduction involves a virus that is cytolytic. It's going to get into the cell, chop up that cell's DNA into a bunch of pieces. Ultimately, the cell blows up, releasing a bunch of virus particles, some of which are carrying cellular DNA with them. So this is just showing the life cycle of a cytolytic bacteriophage, where you have a bacteriophage get in. Actually, it doesn't the whole thing doesn't get in. This is mostly packaging. If we look at a virus particle, real quick like, this is a representation or a model of what's called a T-even phage. This bacteriophage has a fairly elaborate structure. This part up here is called the nucleocapsid. So this cardboard part is the capsid. This is the nucleic acid. This is the only part of the virus that actually gets into the cell. The rest of this is just packaging. So it has a nucleocapsid up here, and then it has a collar and a tail. This tail has a core and a sheath, and then it has a base plate, and it has tail fibers. People want to call those legs. They're not, they're tail fibers, but anyway. This is mostly packaging, and just the nucleic acid gets in. So in the diagram, you see that little squiggle going down into the cell. Nucleic acid gets in there, then it basically, like other types of nucleic acid, it's transcribed and translated, and a bunch of enzymes are made. So the enzymes are going to allow the viral DNA to be replicated itself, and then the enzymes are going to encode the formation of a bunch of viral particles, the capsids, the tail fibers, etc. You get a whole bunch of new viruses built, they're assembled, and then the cell blows up and the whole thing starts over again. So typical cytolytic virus life cycle. Virus gets in, takes over host cell function, chops up the host cell DNA, gets packaged with some of the host cell DNA, and then gets out. If that virus gets into a new cell carrying a little bit of bacterial DNA with it, then the next recipient can become recombinant. However, if the virus stays cytolytic, it's not going to accomplish anything because it's just going to kill that cell too. So again, this is showing some of the host cells' DNA being packaged with the virus. Usually, the virus capsid can only hold so much DNA. So if it's got host cell DNA with it, sometimes lytic genes get left off. Then when the virus gets into a new host, it has to become what we call a prophage. A prophage is viral DNA that goes into the cell, and then it actually incorporates into the host cell chromosome. So here we see this virus. Assume that the virus now, this guy, is one that's carrying its own DNA plus some bacterial DNA from the first host. When that DNA goes into the cell, if it incorporates into the chromosome, this is now called a prophage. So prophage is viral DNA in the host chromosome. And if it does that, then the cell's not going to be killed, at least not right away. And it can stay there indefinitely. That cell will now replicate its chromosome, and it will replicate the viral DNA right along with it. And if that virus was carrying some other bacterial DNA, then that new host is recombinant. And again, it's just going to be reproduced right along with the host chromosome. This is showing the kind of a summary of generalized transduction, which is kind of hard to follow just because it's hard to see one type of DNA from another. But the DNA from the original host is slightly darker. It's packaged up with the virus, and so when it gets in over here, here it is, now in this lighter chromosome. So this cell is now recombinant. If transduction is specialized or specific, this means that only a certain segment of DNA can be transferred from the original host cell. And it has to be, it's gonna involve a virus that is a prophage to start with. So we look at our original bacterial host, the virus is in the chromosome, and it's gonna get out of the chromosome. When it gets out, it can only take a piece of DNA that's adjacent to it in the chromosome. This is why it's called specialized transduction. It can't take any part of the chromosome, it just has to be the part it's attached to. It gets out, it goes to a new host, gets back in, and again, it's gonna have to be, form a prophage. When it gets into the second half, the second host, it has to become a prophage, because if it just kills the host, you haven't accomplished it. So always the final step in transduction, whether it's generalized transduction or specialized transduction, the final step is the virus gets into a new host and becomes a prophage. If it goes into a new host and it's still cytolytic, as I said, that cell is just going to die. And if you're trying to transfer DNA in and have it change the bacterial cell, the cell has to survive. So again, these transfer mechanisms, DNA-mediated transformation, conjugation, and transduction are all involved in horizontal gene transfer. They can occur naturally, but people can also take advantage of this and use these mechanisms to put DNA into cells that they want to put in. Also recognizing that gene transfer occurs, this makes it much, much more difficult for us to try to control bacteria becoming resistant to antibiotics. So one way that they can become resistant is to mutate. That's kind of a chance operation that they might suddenly develop resistance. But if you have one type of bacteria that is resistant, and they can transfer those resistance genes to other bacteria, which they do, then it's not surprising that we have so many bacteria that are becoming resistant to antimicrobial drugs. So yes, it's occurring at an alarming rate. So this is again talking about penicillin. Penicillin is a beta-lactam antibiotic. It was first used extensively kind of late in World War II. Again, Alexander Fleming discovered it in 1928, but it wasn't actually used extensively until World War II. At the point where it was used, it was very effective against Staphylococcus and Streptococcus. It would control most of them. Now, most Staphylococcus are resistant to penicillin. So it's changed a lot. About 80% of all strains of Staphylococcus aureus are beta-lactam resistant. So they picked up those resistance genes. And basically, because we know that bacteria can do these genetic exchange mechanisms, we can use them to our own advantage. So we can use transformation where we can put select genes of interest into cells. That's what we're going to be doing. We're going to transform E. coli. We're going to transform them with genes that we want to put in. We're going to put in green fluorescent protein genes. So you could technically put in any type of gene into bacteria. There is a little bit of a drawback though. As I mentioned, bacteria have restriction enzymes. So if you're going to transfer DNA from some external source, you have to make sure that the bacteria aren't going to recognize that as foreign and just chop it all to pieces. I'll talk about the specifics of this in the laboratory. The other thing, this is talking about making what are called cosmids. So cosmids, they're like plasmids, but they're made from bacteriophage lambda. Bacteriophage lambda is the genome looks like it's a loop, but it's not covalently closed. So you have one strand of DNA and then another strand of DNA complementary. And these little sites here are broken. The only thing that's holding this in a loop shape is this. These things are called cohesive termini. So there's hydrogen bonding here. Otherwise, those things would be separate. So again, it's not covalently closed loop, it's only held by hydrogen bonding. Well, these are big loops, and they have sections in them that have the genes that encode the lytic cycle, which would allow the virus to start taking over the cell and chopping up the host cell DNA and so forth. So basically what researchers can do is take the lytic genes out. Let's say we had a section in here that was maybe this big, it was lytic. If they take those genes out, that DNA still has whatever it needs to get into a host cell and enter into the chromosome, but it can't kill the cell. And so you can replace those lytic genes with whatever genes you want to be studying, and cosmids can be used to transfer DNA into cells. They're cloning vectors. They're just way bigger than plasmids. So plasmids are cloning vectors, cosmids are cloning vectors. So I mentioned cloning vectors when we were talking about plasmid extraction. I said a cloning vector is any type of DNA that can carry DNA into a cell and then replicate it. So they have to have their own origin of replication, so they can replicate inside the host. They have to have restriction sites where the plasmid can be opened up to put in the gene of interest. And the advantage to this over using the PCR to replicate DNA, if you put plasmids or cosmids into cells, they don't just replicate the DNA, they can express it. So an expression vector is something that will carry DNA into the cell, and then that DNA is expressed. What does it mean, expressed? What am I meaning? What am I talking about? Yes, phenotype. But how do you get there from DNA? To express DNA, you're going to transcribe it, make messenger RNA, and then that's going to go to the ribosome and it's going to be translated. The cell is actually going to make proteins. So we are going to get E. coli cells that will express green fluorescent protein. They will make green fluorescent protein. They also make beta-lactamase. This is significant because now bacteria can be used to build proteins such as human insulin. So that's what's shown in this picture. But as I mentioned, there is a problem, a couple of problems. One, bacteria don't have spliceosomes. So you can't take genes for human insulin, take just the gene from human insulin and put it into E. coli. They can't do anything with it because they can't, normally what has to happen, the genes that encode insulin, those genes would encode messenger RNA, and then post-transcriptional modification happens. Introns are cut out, exons are put together. Bacteria can't do that. So instead what researchers had to do was get the messenger RNA from the pancreas, reverse transcribe it into DNA, so they get the actual gene that's going to encode functional insulin. Still, if you try to put that into E. coli, the E. coli restriction enzymes are going to chop it all to pieces. So what researchers had to recognize is that bacteria have these, what are called restriction modification systems. They have restriction enzymes. They also have modification enzymes that will prevent DNA from being cut. You just have to modify the DNA before you put it in there. It took people a long time to figure out how to do it, but now you can put just about any gene you want to into E. coli, you just have to modify it before you put it in. So some human proteins that are currently being produced by E. coli include human insulin, also human interferon, human somatomedin, anyway, C, this is an insulin-like growth factor, and human growth hormone, and several interleukins. So there's quite a number of substances that are produced by Escherichia coli now. And then, of course, although allowing organisms to produce recombinant proteins is beneficial, there are some potentially downsides to this. So bacteria have been genetically modified so they can produce a lot of useful proteins. Genetic modification is also being done to crop plants. And this is really controversial because now we have crop plants that basically, a couple of things, they can produce crop plants that are highly resistant to herbicides, specifically Roundup. So you can buy seeds of plants that are called Roundup Ready, which means farmers can plant these organisms in their fields and they can spray Roundup on them over and over and over again, and the plants don't die. But Roundup is not innocuous. It has some rather toxic substance associated with it, and you just have very high levels of this toxic material on food products now, which is not okay. The other problem with genetically modified food types is if they are genetically modifying certain types of plants to produce insecticides. And although, yes, it does kill the insects that you don't want eating your crop plants, it also kills other insects like honeybees that we're dependent on. And so it's not necessarily a good thing. So I throw that slide in there. Again, it is very controversial business. I'm not going to talk about CRISPR. It's like, no, we've done that too many times. Stop. No more CRISPR. Good, we're at the end. Huh. Well, heck. Well, since we're behind, I should probably start talking about viruses, huh?
Yeah.
Are we behind? Yeah, we are.
You only got 20 minutes.
This kind of goes along with what I was just talking about with transduction. Get on there. There.
Are we going to be lecturing on Tuesday on material that we're going to have on the exam on Thursday?
Depends on if I can get through it this week. So the exam is next week, but it's not a week from Tuesday, it's a week from Thursday. So ideally, we should be finished with all this material this week. The material that is supposed to be on it, which would help if I got started on viruses. So I think the schedule has us finishing viruses even earlier than that. We are a little bit behind because I missed last Tuesday. So let's go ahead and start looking at viruses. No, I'll turn the projector back on. Maybe it won't come.
It looks like this 17 page is 2 lectures.
Wait, what?
Yes.
Oh, Crystal had said earlier that she printed out the notes for this one. It's 17 pages, but I have the schedule here, and it's viroids, prions, human viruses, and viral diseases. So it's 2 days. So
Yeah, we're not going to get through it all. Don't
and
worry.
then this is viruses and viral diseases. It's the last of what's on the exam.
So what has happened in the past, depending on where we are schedule-wise, sometimes viruses and viral diseases get on midterm 2, and sometimes they don't. So we'll see how far we get with viruses in general. It can go either way, it's not a big deal. I just didn't want to be too far behind. I'm more concerned about the lab because we're trying to catch up. Okay. So this is our introduction to viruses with a very brief description of viroids and prions. Sometimes I put it in the beginning and sometimes I put it at the end, so I'm not sure where it is. So again, by the mid-1800s, biologists had come to accept the cell theory. And the cell theory says that the cell is a basic unit of structure for all living organisms. The cell is a basic unit of function for all living organisms. And all cells come from pre-existing cells by means of biogenesis. That's the basic cell theory. There's a bit more to it now because we know a lot more about bioenergetics and metabolism and so forth. So this is just going on to say a more modern version of the cell theory. We know that energy flows, that the hereditary information isn't protein, it's DNA. DNA goes from the parent individuals to the offspring individuals. We know a lot more about the chemical composition of DNA than about other protoplasm, metabolic pathways, and so forth. This is, again, just a review of characteristics of life. So when we get to the final exam, it's good that you remember some of the things we had early on in the semester. Cells reproduce, typically by means of fission, but they can reproduce sexually and asexually. They carry out metabolic processes. Metabolism is both anabolism and catabolism. They grow by assimilation, so they take in materials from the environment, take them apart, and then rebuild them into new cell components. They respond to external stimuli, they mutate, and they have a high degree of organization. So we just talked about mutation a short time ago. So these are the features that make cells living cells, but are cells the only entities that are capable? The reason I'm throwing this out here is because when you start talking about viruses, a lot of references tell you that viruses are not alive. Well, these are the characteristics of life: be able to reproduce, carry out metabolism, respond, mutate, have a high degree of organization. Viruses very definitely mutate. Mutation is purple, but remember when I was talking about mutation, I said mutation is a change in nucleotide sequence of a DNA molecule, but it could be RNA if we're talking about viruses. So viruses definitely mutate. They contain genetic information, either DNA or RNA. So yes, they can mutate. What other kinds of things can they do? They respond. So viruses have to be able to respond to attach to a host cell. They can't carry out metabolic processes themselves, but they often carry genes that encode enzymes, and enzymes are catalysts for chemical reactions. So they take part in metabolic processes. They can basically take over a cell's metabolic process. Do they reproduce? Absolutely. They can only reproduce inside cells, and their mechanism of reproduction is totally different. It's completely unlike what cells do. But still, we've got response, we've got reproduction, we've got mutate. Do they have a high degree of organization? Yes, they do. So they meet many of the criteria of living organisms. They're just not cellular. And again, that dividing line between living and non-living, we kind of made that up. Humans make that up. The abiotic portion of the environment, abiotic is without life. Certain things we recognize, so air, water, rocks, sunlight, they don't have any of the characteristics of life. But entities that are non-cellular but still have some of those characteristics, viruses, viroids, prions, they're kind of in a gray area. And it can be argued either way. Are they alive or are they not? They're somewhere in between. So what are these things? Well, viroids, and here again, watch out for spelling, because we're going to have the term virus, which is a virus. If you have a single virus particle, that's called a virion, and then that's different than a viroid. So a viroid, these are single-stranded RNA molecules. They are not surrounded by a protein coat, so they're just nucleic acid. People recognize these primarily as plant pathogens, but it turns out viroids are fairly numerous. They're all over the place. Here's a diagrammatic representation. It's single-stranded RNA, but these lines that come across in the middle represent hydrogen bonding between the complements as this folds back on itself. So it's a circle that is hydrogen bonding in several places here, and it's also twisted. It's not diagrammed that way because it's hard to see it. So nucleic acid, but it's RNA, a single strand of RNA, except where it is forming these complementary bonds, the hydrogen bonds. They're pretty small, 264 to 375 bases long, and again, no protein coat. This is another diagrammatic representation, again showing quite a bit of double-stranded molecule. So, and this is true of transfer RNA molecules and ribosomal RNA molecules. They also have regions where they have double-stranded nucleic acid with a hydrogen bond. But viroids apparently don't encode any kind of protein. So exactly how they cause disease in plants is uncertain. They're associated with a variety of plant diseases. This is not something you have to remember a whole bunch of different diseases caused by viroids. Just know what a viroid is. So viroids, they're single-stranded RNA that's folded back on itself, and they are known to infect plants and possibly other types of organisms as well. In plants, we know that they cause disease. Apparently, they replicate inside the host cells using RNA polymerase. And some have ribozyme activity. So again, ribozymes are catalysts. They're RNA molecules that have catalytic function. But apparently they don't encode proteins. One of the things that people know about viroids, because they exist independently, they're not inside cells, they're not surrounded by a protein coat, they show that RNA can exist in the environment, and biologists think that probably the first type of nucleic acid to exist was RNA, not DNA, and that all DNA was encoded from RNA at some point. The next type of non-cellular entity I want to talk about are called prions, or prions, depending on who's pronouncing it. These are proteinaceous infectious particles. So they are proteins, but they're capable of infecting animals and causing disease symptoms. This is another type of entity. There's quite a bit more known about these now, and apparently they occur naturally within some types of fungi as well. Anyway, these things are usually transmitted by being ingested. So they can be in the environment, a host organism will take them in by eating them, and then what they do is migrate into the brain, and they cause damage to brain tissue. They can also get in through tissue transplantation, contaminated surgical instruments, etc. And there is one type of prion, or prion-like protein, that is genetically transferred along certain family lines. So prions are proteins. They were first identified in 1982. Stanley Prusner at UC San Francisco is recognized for identifying these things, and it was very controversial at the time because people thought there had to be nucleic acid associated with these things. You can't just have protein, you have to have nucleic acid. And they searched and searched, but apparently they're just proteins that are infectious agents. They do not have nucleic acid. He was studying a disease of sheep called scrapie, and it was called scrapie. You can see these sheep are missing wool on parts of their bodies. This is because the sheep lose their muscular coordination. They begin to suffer from nerve damage, and they would fall against the rock walls and scrape their wool off. So scrapie was kind of a diagnostic feature that they had prion disease. So they're scraping their wool off. Well, what he found out was that the protein that caused scraping is a very stable protein. So in the environment, this protein can withstand heat, radiation, etc. Its structure is extremely stable. It took some time to figure out that this protein is actually identical to a normal brain protein. So we have genes within our brains, we have genes within some of our cells that encode proteins that can become prions. It's just that the prion is a different folding. So when we were talking about proteins early in the semester, I talked about primary structure of the protein, which is amino acid sequence. Secondary structure of the protein is how they're folded or twisted into a helix. Apparently it's a change in secondary structure that makes a difference between a normal brain protein and a prion. The prion folded protein type is much more resistant to damage, much more stable in the environment. But it has the same amino acid sequence as the normal protein. So again, the gene that normally codes proteins that can become prions, that gene is normally present within nerve cells. I'm not sure that they know exactly what that protein does, other than it produces this protein that can potentially be turned into a prion. Well, why does it matter? Because prions cause what's called transmissible spongiform encephalopathy. So it causes brain tissue to become sponge-like, causes the formation of plaques, these little holes throughout the brain tissue, and if that happens enough, the brain doesn't function properly at all, and the individual will die. So scrapie is just one disease. That's a disease of sheep and goats that is associated with prions. Common names for these spongiform encephalopathies: if it's in cattle, it's called mad cow disease. If it's in elk, deer, moose, it's called chronic wasting disease. If it's in mink, it's called transmissible mink encephalopathy. In other words, it's fairly widespread. These prions are in the environment, and again, because they're very stable and because they enter into their host organism through ingestion, it's fairly easy to pick these things up. This is some brain tissue that has been damaged. Well, so exactly what happens? Why, if these are proteins, they're not encoded for by nucleic acid, how does a protein reproduce? Well, it turns out they don't actually reproduce. Instead, what they do is they get into brain tissue and they cause normal proteins to change their secondary structure and become prions. It's a recruitment process. So if they get into the vicinity of a protein that has the same shape, the same amino acid sequence, they will refold that protein and turn it into a prion. And again, as I said, once they're in prion form, they're extremely stable. A little bit scary, actually a lot scary, if they get into the human food supply. In humans, there are a number of diseases that are recognized as being caused by prions, and different names for these probably because they were discovered in different peoples throughout the world. So there's Creutzfeldt-Jakob disease, which is CJD, Kurtzman-Straussler-Schranker syndrome, GSS, Albert syndrome, which is in infants, and as I said, fatal familial insomnia, which is genetic. So people have genes that encode these proteins that tend to turn into prions by themselves. And Kuru was first identified in tribal people in New Guinea. And apparently one of the customs of the people was to, when an individual died, family members would gather and they would consume the brain tissue from that individual to preserve memories. Unfortunately, they were also preserving prions. So prions got into the population, and then they were kind of maintaining prion disease by passing prions down family lines, which is devastating. So again, typically you would acquire these by ingestion. If you eat the prion proteins, they can survive going through the stomach, through the intestine without being damaged. They get into the bloodstream, they migrate to the brain, and there they cause normal brain proteins to become prions. They can also get in through tissue transplantation, use of surgical instruments, etc., etc. So it's a totally unique method of reproducing, where again, what they're doing is just getting into the vicinity of normal proteins and causing normal proteins to become prion proteins. It's a recruitment operation. And once you have a bunch of these abnormal proteins, they begin to cause the formation of amyloid plaques. They basically destroy the brain tissue. These things are highly resistant to damage by heat, pressure, radiation, etc., which is very concerning because we know that certain animal tissue contains prions, and we ingest a lot of animal tissue. I'm not real keen on eating certain animal tissues anymore. Primarily beef, but also moose, deer, elk. It's like, we have chronic wasting disease in the US. And whereas the FDA used to monitor for mad cow disease, prion disease in the US very stringently, I remember distinctly watching a newscast where somebody said, well, the FDA was going to stop testing because they didn't want to cause a public-wide panic. And I thought, wait, you know there could be prions in our food supply, but you don't want to upset people, so you're not going to test for them? Yeah, that's scary. That is just scary. So, and again, if they're in the food supply, it's really difficult to process them out. We process a lot of meat. You get this pink goo that gets put into hot dogs and, slabs of this and that and put on sandwiches. It's like, could you kill prions by these various methods of treating meats? And probably not, because they're very resistant to radiation, heat, toxic chemicals. Apparently, people that work in labs where they're working with prions, to get rid of these things, they have to autoclave them many, many times to get them to denature enough that they are no longer infectious agents. So they're extremely resistant to damage. And more widespread than we even can imagine. I eat a lot of bird. Surprised I know birds don't have prions. So infection is almost always fatal. It's going to take a while, but eventually it causes enough destruction of the brain that it kills people. So people are trying to come up with therapy, you know, what can you do to stop it? Stop that refolding process. So they have a vaccine that prevents prion disease in mice. Maybe eventually it will prevent prion disease in humans. Pretty scary stuff, those prions. Oh look, we didn't even get to the viruses. Oh, I thought this was interesting. Apparently in Japan, they genetically altered cattle to produce cattle that didn't have the protein that could be used to make prions. Which really makes you wonder, what's the function of that protein in a normal brain? If you can genetically modify animals so that it's not there, and they still function. Then again, how functional is a cow? I don't know. Certain things they just don't do. Okay, so we'll start there next time. Introduction to viruses. We still might not get through everything, but maybe.